Hi, everyone. Welcome
Hola a
to
todos y
our research overview of genetic therapies in CF. I'm so glad that you're here. My name is Medora Fry. I am an adult living with cystic fibrosis, and I was actually diagnosed late at age 18. I am particularly interested in what both of our speakers have to talk about today because I'm currently not on Trikafta, even though I am eligible due to side effects. So just like probably most of you, I'm extremely excited about what our presenters have to say. So thank you so much again for being here, everyone. We have a couple housekeeping items to go over. So to turn on the live Spanish translation, you can click on the interpretation button in our Zoom menu. We have closed captioning that's also available, and you can turn the subtitles on or off by clicking the CC icon at the bottom of your screen. The event is being recorded and will be posted on YouTube. Everyone watching tonight will receive an
todos
email
los
with
que
a
estén
link
viendo esta noche van a
to
recibir un
that
correo
recording.
electrónico para
If
esta
you use the event
grabación.
chat,
Y
you
están
can change it
usando
to
el
all
chat,
panelists and
pueden
attendees so
cambiarlo
that everyone
a
can see
todos
your
los
comments. And the default is to panelists only, just so you know. And we'll have time at the end to take your questions. We'll be doing a Q&A, and you can actually type your Q&A questions into the box at any time.
y
We
respuestas
won't
y
be able
también
to
pueden
answer the
poner las preguntas al
questions
final,
about like
las
individual
preguntas que
diagnosis
no
or treatment plans.
podamos
So we encourage you
responder.
to use the info that you do learn today to have
Les
conversations with your care
pedimos
team.
que después
Our
tengan
agenda
estas
is that
conversaciones
we'll
con
just
su
have our
equipo. Este es
two
el programa que tenemos
presentations, and
para
then
el día de
afterward
hoy.
we will
Tenemos
have
dos
that Q&A.
presentaciones y después vamos
So
a
I'm
tener una
going
sesión de
to
preguntas
first
y respuestas. Así
introduce
que
Kara
primero
Foshay. She
voy a
resides
presentar
and is
a
a native of Northern Virginia. She received her Bachelor of Science degree in biology from the College of William and Mary, her PhD in cell biology from the Department of Human Genetics at the University of Chicago. Kara joined the CFF 4 years ago and is a leader on the science team, where she manages the industry-based genetic therapy portfolio. Outside of work, she enjoys food, yoga, and life with her husband, 2 kids, Bella and Ryan, and dog Ziggy. So welcome, Kara.
Thank you so much, Medora, and thank you all for attending. I am thrilled to be here tonight. Let me go ahead and share my slides. All right, so hopefully you can all see these slides, and I'm here to kick off this genetic therapies overview. I'm sorry, I'm adjusting my screens. I'm not at my normal desk. Okay, so here we go. The flow of genetic information is really important for understanding how mutations in the CFTR gene can cause CF. And it's also really important for understanding how genetic therapies can benefit people with CF. This flow starts with DNA. All of our cells in our body contain the same DNA. And that's where our genes are found. Genes are transcribed or copied into mRNA. mRNA is then translated into protein, which is folded and becomes the functional output in our cells. This is kind of like a food blog. So I imagine many of you who like food, like myself, have gone to a food blog and you start reading and there's a ton of information. And it's actually pretty hard to find the information you want. So you can think of that blog and all of its information as the genome. There's a lot of important information, but there's a lot of information we don't always need. So when you scroll through and you highlight and you write down your shopping list, that's a little bit like transcription into mRNA. mRNA has the key bits of genetic information that your cells need to make a protein. You can think of cooking the food and serving it on your plate sort of as translation and making your protein. Your cheesesteak, in this case, is the functional component that works in your cells or in your stomach. So, as many of you know, because you're an educated audience, CF is caused by mutations in the CFTR gene. and there are many different CF-causing mutations. The point of the picture on the left is to demonstrate that CF-causing mutations can occur throughout the gene, and it's a really big gene. But the location of where a mutation is in the gene will dictate how it affects the CFTR protein. The CFTR protein is depicted on the right, and some parts of the protein are more essential for its function as a chloride channel than others. So mutations in those regions may produce a more severe case of CF than people with other mutations. One example is G551D, which we call a gating mutation. The mutation in the DNA is copied into the mRNA when it's made, and it's present in the protein when it's made. In this case, the protein doesn't open and close correctly, so it doesn't function as it should. Following our food blog metaphor, imagine we delete the section that says thin sliced ribeye steak. We still get a recipe list or an mRNA, but it's missing something. And we still get a sandwich at the end, but it's really missing something. And it doesn't function the same way as a cheesesteak. Now, when a dysfunctional protein is made as a result of a CFTR mutation, modulators can act on that protein to help restore the function. In the case of a misfolded protein, correctors like tezacaftor can improve protein function. In the case of a gating mutation like G551D, potentiators can help the protein to open so that chloride can flow. It's important to note here that the mutations that are present in the DNA, the RNA, and the functional protein are still there. The modulators simply help the protein to function. However, some mutations can prevent any CFTR from being made. And patients with these types of mutations cannot benefit from modulators. Again, following our cheesesteak metaphor, a premature stop or X mutation, which many of you have heard of, looks like a stop sign in the DNA, in the RNA, and no protein is made. And therefore, we have an empty plate. Without a protein for modulators to work on, they can't exert their beneficial effects. But when no protein is made, genetic therapies can help. Genetic therapies are therapeutic approaches that fix the DNA, perhaps via gene editing, or replace the DNA via gene therapy or gene transfer, or RNA via RNA therapy. And it's important to point out that genetic therapies are actually quite different from modulators. Modulators are small molecules. And right here, I show a schematic of what the chemical structures of Trikafta look like. There are 3 different drugs, and they're pretty tiny in terms of size. Next to it, I have a picture of 1 nucleic acid in the CFTR mRNA. But CFTR mRNA is actually 6,200 nucleotides thereabout. And you can see that it's much, much bigger than 1 molecule of Trikafta. You could compare this to a drug the size of a kumquat compared to a drug the size of a watermelon. A drug the size of a watermelon would be a lot harder to handle or to deliver. And in fact, genetic therapies can't diffuse through your cells like small molecules can. The nucleic acids they're made up of are big and bulky and also fairly sensitive. So they have to be packaged in what we call a delivery vehicle. You may have heard us talk about some of these in the past. and heard us talk about things like a viral delivery vehicle or a viral vector, like AAV or adeno-associated virus, or a lipid nanoparticle or an LNP. These are protective covers that help a genetic therapy get to the cells that it's targeting. And it's important to note that genetic therapies must be targeted to specific organs and taken up by those cells for them to function. Now, we know that CF is a multi-organ disease, but because of this targeting aspect, genetic therapies will start in the lung. We hope that eventually they'll be able to help all organs affected by CF. Before I jump into what these therapies might look like when you take them, I thought I'd take a minute to focus on our pipeline and what the drug development process looks like. At the top of this funnel, you see the word discovery. We often refer to very early drug development programs as discovery stage. And this means they're still thinking what a drug might look like. They're still building it. Today, we have a lot of gene editing therapies in our discovery stage pipeline. These are early and pretty far from the clinic for CF. In the preclinical section, these are programs that are trying to say, how effective could I make my drug in CF models? Could it work for CF? Here, we have a lot of mRNA therapy programs and gene therapy programs, not as many gene editing programs. These are closer to the clinic. And finally, we have programs in the clinical trial section, and these are currently enrolling for CF. We'll touch on these later. It's important to note that currently there are no approved genetic therapies for CF, and that the average time from discovery to market for any drug in any indication is usually estimated at 10 to 12 years. So this is a long runway for these drugs to get to all people with CF. So how can genetic therapies help? We'll start with gene editing, which I mentioned is the furthest from clinical trials and furthest from the clinic. Gene editing is an actual change to the CFTR gene in your cells. The proteins that are delivered in a gene editing therapy will essentially erase and replace your CF-causing mutation in each cell that they're delivered to. As a result, mRNA that will be expressed from your gene will no longer have your mutation, and the protein will function as normal. The cheesesteak will be restored. So what would a gene editing therapy look like? It would target the basal stem cells and the differentiated cells of your lung with editing proteins that would change the CFTR gene in your cells. It's important to note that by targeting the stem cells, which continually renew and replace old cells, gene editing offers a permanent solution for CF. I didn't say cure because these initial therapies will target the lung only, and CF is a multi-organ disease. To be a cure, these therapies would have to correct the CFTR gene in every organ and every cell affected by CF. We think gene editing is particularly exciting because only a few months ago, the FDA approved the first gene editing therapy in the United States, Casgevy for sickle cell disease. Now, this is a little bit different than gene editing therapies for CF because the editing took place outside of the body in cells that were then transplanted back. However, it still represents clinical precedent for this type of therapy in a genetic disease. There are challenges and risks associated with gene editing. And one of these is that the technology is still really early stage. These trials aren't expected for years, and so we still don't fully know what the risks might look like. We don't know how editing one part of the genome might affect other parts of the DNA, and thereby the RNA and the protein and cell function. We also worry about unintended changes in the CFTR gene or in other genes. So this type of therapy is not without risk. The good news is the field is being driven by leaders and it is evolving really rapidly. And so we continue to learn more every day. It's really an exciting field with fast progress. Next, we have gene therapy or gene replacement. In this type of therapy, no changes are made to the actual genome. Instead, a new copy of CFTR is delivered into your cells. Your mutated DNA will still be there, but the new copy will be transcribed into mRNA that doesn't have a mutation. It will then be translated into protein that doesn't have a mutation and can function. So what would a gene therapy look like? This would target the differentiated cells of your lung. It could touch or hit basal cells, but the important part is that it's taken up by the cells that would normally express CFTR and therefore could restore function. The duration of this type of therapy is really unknown. We expect that the CFTR DNA would last for the life of those cells, but as the cells get old and die and are replaced by new cells, the therapy would go away and it would eventually have to be redosed. We expect this period of time to be months to years, but it'll take successful clinical trials before we have an exact time frame nailed down. Gene therapy is exciting because there's a lot of clinical precedent here. There are AAV therapies for SMA like Zolgensma and lentiviral therapies like Skysona for CALD. And we have a CF trial enrolling now. 4D Molecular's AAV trial for CF is open and enrolling. There are challenges to gene therapy as well as gene editing, which I highlighted earlier. These include rare but unwanted integration of the viral genome material. The clinical significance of this isn't known, and the occurrence is very, very rare in model studies, but it's something that we hope you're all aware of. Another challenge here is the inability to redose. It's possible that the immune system can recognize viral therapies as sort of a vaccine. It can silence the expression of the new gene or prevent the virus from being effective when it's redosed. Studies to examine how redosing might work are still in progress. The last type of genetic therapy I'd like to highlight is RNA therapies. And this could be short nucleotide therapies like ASOs or mRNA therapies, which you may have heard more about. These type of therapies skip over the DNA step and instead introduce a new copy of non-mutated CFTR mRNA. The mRNA is translated into protein, and therefore you have a functional CFTR. What would an mRNA therapy look like? It would target, again, those differentiated cells in your lung epithelium that are replaced over time. mRNA therapy would likely target the lung and be delivered as an inhaled therapy. It's less likely to become a systemic therapy, even in future versions, and that's because mRNA is expressed in any cell that takes it up. There's concern that mRNA for CFTR in the wrong cells could have detrimental effects, and therefore we only want to deliver it to organs where we would want CFTR. We know that mRNA is short-lived. it only lasts for a couple days in your body, and so frequent redosing will be necessary. mRNA is exciting in terms of clinical precedent. I think most of us have now had an mRNA therapy in the form of our COVID vaccine. So we know that these types of therapies can work and can be safe. We have ongoing CF trials for mRNA encapsulated in lipid nanoparticles, or LNPs, from Vertex Moderna and also from Arcturus. These therapies are transient, they're reversible, and no permanent genetic or cellular changes will result from these types of therapies. The challenges and risks include immune stimulation. Inhaled LNPs may have side effects caused by the immune system, such as fever or lung inflammation, and frequent dosing associated with these side effects could make it hard to tolerate. I also highlighted that these aren't likely useful for the whole body because daily or every other day IV dosing is really not practical, and unregulated systemic CFTR could have side effects. So because we have trials in the clinic now, we know a little bit more about what these gene therapy trials might look like if you choose to participate. And I know this is a super busy slide. You can email research@cff.org if you'd like to get a copy of it. And I'll also dive in to a few key sections and highlight some information that I think may be particularly important. So on the left, we have CFTR gene therapy. So introduction of a new copy of the CFTR gene. On the right, we have CFTR mRNA therapy. Introduction of a new non-mutated mRNA for CFTR. So who would be eligible? What mutations? The good news is that both of these types of therapies would be amenable to any mutation, rare mutations, splice mutations, modulator ineligible mutations. These types of therapies will enroll patients with those mutations. mRNA therapies, especially those in early studies only looking at safety, may also enroll modulator eligible patients. Again, not necessarily looking for efficacy, but looking for safety. In terms of the route of administration, the trials that are ongoing now are focused on the lung, and it will be local nebulized delivery through an inhaled mesh nebulizer. So in this case, mRNA therapy and DNA gene therapy will be delivered in the same way. When we compare the duration of effect, you see that these are different. As I mentioned before, gene therapy, delivery of DNA, is expected to last for months to years, but we don't know exactly how long. mRNA therapy will only last for 1 to 3 days in the body and will have to be redosed frequently. So finally, I thought I would highlight some key considerations. We know from recent press releases that 4D Molecular has reported 1 patient with severe lung inflammation in their AAV trial. Side effects like this are something we want the community to be aware of so that they can be truly informed when they choose to participate in a clinical trial. The potential for immunization against future doses of the same therapy is another issue that I highlighted. And these trials will require a high number of clinic visits due to drug administration, which will be done in clinic, safety assessments, and a long-term follow-up. When we look at mRNA trials, there are also some risks or some key considerations. These include risks that were evident in the earlier Translate Bio mRNA trial, which included exacerbation, fever, and in some cases, nausea and vomiting. These trials will also have a higher number of clinic visits due to drug administration in clinic, safety assessments, and follow-up. At the bottom, I highlight trials that are currently enrolling or close to enrolling here. you see that 4D is currently enrolling. Crystal Bio, which is not supported by the TDN, is enrolling in the US. And Boehringer Ingelheim and Spiravant are programs that are in the late preclinical stage or nearing the clinic. So there will be several gene therapy trials for CF ongoing soon. In the mRNA space, we have Translate Bio, which was safe. Fevers were noted, but there was no FEV1 benefit, and that program was terminated. Currently, Vertex Moderna is enrolling in their mRNA LNP trial in the United States. Arcturus is enrolling in New Zealand. And I believe today Recode put out a press release citing that their trial was just about up and running currently. So we will also have several mRNA LNP trials ongoing. And with that, I will end. Thank you very much for your attention, and I will pass it on to the next presenter, Katie Howe.
Perfect. Thank you so much, Kara. I am extremely excited to get to the questions in regard to your presentation, especially maybe, what different bodily systems in the future some of these therapies will be potentially available for, such as the pancreas, GI, since right now everything is lung targeted. So yeah, I'm super excited to get to the Q&A as well. But first, I guess not first. Secondly, we have Katie Howe, who is a native of Michigan and received her Bachelor's of Science in Nutritional Sciences from Michigan State University and her Master's of Science in Human Nutrition from Winthrop University. She resides in Raleigh, North Carolina with her husband and 2 dogs, Wally and Penny, and is an avid trail runner. So Katie, I will let you take it away here.
Thank you, Medora. Let me just go ahead and get my slides up. Yes, thank you, Medora, for the introduction. Thank you, Dr. Foshay, for that wonderful presentation, and thank you all for being here this evening. I'm excited to be here to discuss the logistics of participating in a genetic therapy clinical trial and the resources available to learn more about clinical research. Just a quick note, as we move forward throughout this portion of the presentation, the links for resources mentioned will be added to the chat so you can follow along and save them for future use. As the clinical trial navigator role is new to the CF Foundation, I'd like to first discuss what it is and how it can be beneficial to you. So who is the clinical trial navigator? It's me. I am a family member of people with CF, as well as a friend of people with CF and a former CF research coordinator. My role is to help you understand the clinical trial process, answer questions about trial participation, assist in using the clinical trial finder, and to facilitate contact between you and a research site. You can contact me at this email address, trialnavigator@cff.org, and you can expect to hear from me within 2 business days. As I mentioned, one of my roles is to explain the clinical trial finder and how to use it. It is a very resourceful tool when looking for clinical trials to participate in and to learn more about what trials are being conducted at this time. You can find the clinical trial finder at this web address, and there are multiple ways to use the clinical trial finder. You can utilize the tutorial video found on the clinical trial finder website, or you can contact me and I can help walk you through the process. This is just a quick view of the what the clinical trial finder looks like. When searching for trials, you can search by age, location, or mutation. You can find trials that are currently enrolling or view study results. You can contact a research coordinator at a specific site to express interest in a specific trial, and you can sign up for email alerts to be notified when new trials are posted. Some additional resources available are the genetic-based therapy resources found on cff.org. These include Understanding Genetic Therapies, which describes the basics of CF genetics, as well as the difference between mRNA therapy gene therapy, and gene editing and how they could work for people with CF. Also, there's a genetic therapies for CF glossary of key terms. These are terms regularly used when discussing genetic therapies. Further, there's questions to ask before enrolling in a genetic therapy trial, which are questions that you can ask your provider, care team, or research team when considering participation. All of these resources can be found in Spanish at the below address. Next, because I know there's a lot that goes into the different phases of clinical trials, I'd like to go over those before we dive into the logistics of clinical trial participation. As we know, clinical trials build on each other and direct new research. During phase 1, the duration is generally short, lasting only a few months. The number of people participating is quite low, anywhere from 20 to 100. And the focus of phase 1 trials is safety in determining an effective dose. During phase 2, the duration is a little bit longer, ranging from months to 2 years. The number of participants increases to several hundred. The focus is efficacy, which is the power to produce a desired result, as well as side effects. Moving into phase 3, the duration is even longer, ranging from 1 to 4 years. The number of participants increases drastically, and the focus is efficacy and monitoring adverse reactions. The last phase is phase 4. This is typically occurs after FDA approval, lasts for several years. The number of participants is quite large, and the focus here is post-marketing safety and efficacy. I mention all of this because we are here in phase 1 of genetic therapies, meaning there will be a smaller number of people with CF enrolled in these clinical trials, and we have a ways to go before they will be available more broadly. Also, during these phase 1 trials, typically only adults are enrolled at this time. Currently, this is where the CF Foundation Genetic Therapies pipeline stands. As you can see, those with a star next to them are currently enrolling, and others have yet to begin enrolling or are in the preclinical phase. Referring back to Dr. Foshay's slide about what will a CF gene therapy trial be like, I'd like to talk more about the logistics of participating in a genetic therapy trial, focusing on what to expect regarding the length of trials, the number of visits, dosing, assessments, travel, reimbursement, challenges, safety, and risks. So what to expect? On average right now, the length of genetic therapy trials ranges from 6 months to a year. The number of visits can range anywhere from 8 to 11, and those are for those trials currently enrolling, but this will differ dependent upon the clinical trial. The length of the visits can be anywhere from 3 hours to multiple overnight inpatient stays. For dosing, as Dr. Foshay mentioned, for mRNA trials, this could potentially be daily or every 2 to 3 days. And for genetic therapy trials, it would be a single dose. Again, determining an effective dose is part of a phase 1 trial, so daily versus every few days is being determined. If participating in a clinical trial, some of the assessments that may be conducted include vitals, height and weight, blood draws, ECG, spirometry or PFTs, the possibility of bronchoscopy and chest x-rays, sputum induction, and MBW or multiple breath washout. This measures the efficiency of gas mixing in the lungs, and it can be an outcome to detect early lung function impairment. One of the major questions we're asked often is about travel and reimbursement. Travel is generally arranged by the research site and the sponsor, and reimbursement for travel, meals, and completion of study visits is dispersed after signing a consent form and after each study visit. Some of the challenges we're hearing about is that the time commitment is quite long. Because they are phase 1 trials, the sponsors may change the structure of the trial, including the length of the trial, the number of visits, what the visits may include, and in addition to which assessments will be conducted. Right now, some of these trials offer home health options where some of the visits can be conducted in your home. However, this differs from research site to research site. So at some sites, all study visits would have to occur in the clinic if they don't allow home health options, whereas other sites, it can be a combination of in-clinic visits as well as in-home visits. When it comes to safety and risks, we know that there will be long-term follow-up via the FDA. Right now, significant safety issues are not anticipated, but as always, safety is a priority in every clinical trial, but we recognize that we are in new territory. Therefore, safety is even more important during this time. Another safety measure is that enrollment for these trials is quite slow, so patience is key right now. When it comes to risk, as previously mentioned, there will be multiple clinical visits for drug administration, safety monitoring, and follow-up. We have seen fever and exacerbation and the potential for immunization against future doses of the same therapy. The safety resource you see here on the right can also be found on the CFF website and will also be located in the chat. And this is for all clinical trials and discusses what safety measures take place before enrollment and throughout the study. Next, I'd like to discuss what we're hearing about genetic therapy trials from the community. The CF Foundation reached out to community voice members to better understand the barriers to participating and the challenges for those not eligible or benefiting from modulator therapy. Some of the key themes we have heard is that there's a lack of engagement in conversations about genetic therapies by care team members. There's an unfamiliarity with the basics of genetic therapies and how to find a clinical trial. And there's a need for clear, more accessible resources on the CFF website. You can join Community Voice to inform CF research projects, shape priorities, and make your voice heard. And you can learn more at the web address below. Lastly, I'd like to mention how you can become involved. If research is being conducted at your CF center, you can use the genetic therapy questions to start a conversation with your provider. You can ask to speak to your research team. If it's participating at a different CF center, other than where you get your care, you can use the clinical trial finder to search for genetic therapy trials that interest you or are available in your area. And you can talk to your provider about participating in these trials as well. For general information about participation, or you need help with getting involved or participating in a trial at your center or a different CF center, you can contact me to learn more about genetic therapy trials and the resources available. Additionally, as I just mentioned, you can help shape future research projects by joining Community Voice to make your voice heard and ensure CF research meets the community's needs. This concludes my portion of the presentation, and we'll begin the Q&A session. So I will hand it back over to Medora and bring back on Dr. Foshay. Thank you so much for your time.
Great. Thank you so much, Katie. And I do just want to extend once again a thank you to both Dr. Foshay and Katie for their insightful presentations. I've been extremely excited about everything that they've been talking about tonight. And I just want to also thank them both for the work that they do for the CF community. Obviously, research is incredibly important, and a lot of us may, like myself, may not even be here if not for the work that they do. And also, I know Katie's position, I believe, is kind of new, and so I'm excited, Katie, to have you as a resource for the foundation. So thank you both again. We are going to get to the Q&A now. Let me just try to pull up all my stuff here. Okay, so our first question, and I think this can be for either of you, is what is in the pipeline for those who cannot take modulators or those with rare mutations? So I know that we kind of answered this, but is there anything, I guess, further that you'd like to add or just briefly go over?
Yeah, that's right. So the great thing about genetic therapies, especially gene therapy and mRNA therapy, is that they will be targeting modulator ineligible people with CF in their trials. They will eventually, if approved, be able to help anybody with CF, but the initial focus will be on people who are ineligible for modulators or who have rare mutations.
Okay. Let's see. I'm so sorry, everybody. My Q&A thing is not updating here, so just give me 1 second. Okay, we're back in business. Sorry about that, everybody. So how will genetic therapies impact patients with lung transplants or multiple organ transplants?
I'll go ahead and take that one too, or at least kick it off. Since gene therapies are targeted to specific organs and replace mutated copies of CFTR, they really won't benefit people that have received a lung transplant. Lung transplant recipients have new organs with new cells that have unmutated CFTR. So they really wouldn't benefit from these types of therapies. The same would go for multiple organ transplants.
Perfect. Okay, our next question, once again, I'm not sure who would be best fit to answer this one, but what is a realistic time frame for these therapies making it to patients? And additionally, when could they be available to children?
I can take that one, Kara. I think as we mentioned in both of our presentations, that these are very early phase trials, and from discovery to market, it takes anywhere from 10 to 12 years. Additionally, these are phase 1 trials, and they do start off with adults first. And so the likelihood of them becoming available for children is a little ways off, and including FDA approval.
Are we expecting that future stages of the genetic clinical stages, will accept participants with lower lung function? This person is saying one of their biggest hurdles for participating is not having a high enough FEV1 to meet eligibility criteria.
We do typically see that there is a specific range for eligibility criteria. That it is very dependent upon sponsor and trial, but we do want to ensure that the people that are participating in these trials are capable of moving forward with the trial throughout the entire length and are able to maintain their health as well.
Why are trials for gene editing not expected for years?
Yeah, that's a great question. The reason is that this is really new technology that is just being developed and being studied in academic research labs. As I mentioned, we've only just had the first FDA-approved gene editing therapy in the United States earlier this year. And so it's really just in that discovery stage. And as we highlighted in the pipeline and from discovery to market, this is a long process of validating what a drug might look like, validating that it could work in CF, and then testing it in clinical trials. And so unfortunately, it's a slow process. But the good news is it gives us time to really identify the risks and benefits of this type of therapy.
Great, yeah, and I'm sure, I know for myself, obviously safety is a, huge concern. So I do appreciate, although obviously we would love to have things, as quickly as possible and instantly, I think taking the time, to make sure things are safe and whatnot is obviously super important. Our next question is, could you please comment on clinical trials for ASO therapy?
I can take that one if you want to start. So SpliceSense is a company in our pipeline, and they're developing an ASO therapy for 3849 plus 10 kb. So this is a mutation-specific therapy. They recently completed their phase 1 trial in healthy volunteers and are looking to start their phase 2 trial in CF in the first half of this year. So stay tuned.
Okay, and I think another kind of follow-up question to that is, if that ends up being successful, how quickly would they be able to rework for another splice mutation? I think that's related.
Yeah. So unfortunately, if they turn to focus on another splice mutation, that would be a new drug, and it would have to go through the same regulatory path that others go through. So it would still be, several years to develop and test before it would be available to patients.
Are researchers using AI to shorten the timeline of 10 to 12 years?
I don't think that can be shortened I don't want to say it can't be shortened. Definitely researchers are using machine learning and AI guided algorithms to develop new therapies, but that's really in that early discovery stage. So potentially identifying more potential therapies, they still have to go through the same preclinical development and clinical development pipeline. Again, that's for our own safety. We want drugs that are both safe and effective. So I don't think that AI anytime soon will be shortening that.
Sure. Okay, is there any thought on how effective mRNA and gene therapy will be compared to Trikafta? And additionally, will any of these be done in tandem, like someone taking both Trikafta and mRNA, for example?
I mean, these therapies are in trials, and that's what the trials are for, to determine safety and efficacy. So I don't think we can compare directly. One thing we know is that these will target only the lung. So people who are on Trikafta and receive benefit to their GI system or pancreatic function, that won't happen with mRNA or gene therapy, because these will be lung only. One other concern, as we've mentioned, is delivery. So these are an inhaled delivery, and the delivery vehicle has to be taken up by all the cells. And we know that mucus in the CF lung is a major barrier. So if uptake is not as good as modulators, the response may not be as good. But we really need those clinical trials to tell us whether uptake is good enough to see clinical benefit.
Interesting, thank you. I think the next one that I also found kind of interesting would be how soon or in what phase can one of the current phase 1 trials of gene therapy medications be available for compassionate use by the FDA?
Again, these are early phase trials, so for discovery to market is still 10 to 12 years. And so even for compassionate use, when you get into these phase 3 trials before FDA approval, you're still looking at several years before that's a possibility.
Sure. Can you summarize what results are published from DNA and mRNA phases completed?
Yeah, so I can start. The 40 molecular DNA gene therapy trial that's ongoing doesn't have any published results yet. You can Google, and there are some preliminary results that are available through press releases, but I don't believe there's any publications. The Translate Bio study that was completed, there is a scientific publication that goes in depth on the results there. And I guess if somebody wants to read that, they can email me and I can send it. But unfortunately, that study did not detect any improvement in FEV1, and so the program was discontinued.
Our next question is, are some patients going to be given a placebo treatment? And I'm assuming that's just in all gene therapy trials. I'm not sure exactly what they're referencing, but or which study specifically.
Yeah, Kara, you can correct me if I'm wrong, but as of right now, the ones that are currently enrolling do not have a placebo arm of the trials.
Yeah, I think
do you expect others to?
Yeah, I think for mRNA trials, there may be placebos. Probably for viral-based gene therapy, there won't be, and I would think in editing trials, there may not be. So you may see different designs than we saw for modulators. It certainly was much smaller placebo groups.
Okay. What is the number of patients that aren't currently eligible to use modulators?
If I can remember this number, and Kara, you can, it's about, in terms of eligibility, it's approximately 10% of the population. And so I know that that is around, I believe we said around right under 2,000 patients in terms of not eligible for modulators. but that does not include those who are eligible but aren't able to take modulator therapy.
Definitely. Okay. Next question is, what mutations are eligible for the trials enrolling, and how do you find this information? We might have kind of answered that one, and I'm sure there is a link in our chat, but if either of you just off the top of your head quickly knows that. Maybe it should be all, but
Yeah, you can find this information, the clinical trial finder, and that will also give you information about eligible mutations, but it's typically those who are not eligible for modulator therapy or those not benefiting from modulator therapy, excuse me.
okay. I love this question as well. What are the lessons learned from the previous attempts at genetic correction in CF?
Yeah, I can take a stab at this. So we know that there have been previous attempts. There's actually a pretty long history of attempts at gene therapy in CF. And what I can say is that we've learned a lot more about delivery. And this is, I think, maybe my second or third question where my answer has been about delivery, but we really see this as a key challenge to successful genetic therapies. So these have to be encapsulated in their delivery vehicle. They have to get to the target cells and taken up by it. So there's several steps. We have learned since the previous realm of genetic therapies a lot more about which cells normally express CFTR and which cells we need to target. And we've learned a lot more about how to target these therapies, how to make them, how to control which cells that they go to. And so we're really hopeful that this time around delivery will be more efficient. And that's really going to be the key, getting as many cells as possible that would normally express CFTR to take these up and express the new copy.
What do you think are some of the most important concepts for patients to understand when discussing gene therapy? This might be a good one for Katie.
Yeah, I think the biggest thing is patience. We are, the number of participants in these trials is very small for a reason. Again, going back to the safety aspect of genetic therapies and conducting these clinical trials, I know that and just making sure that you are utilizing these resources, educating yourself as much as you can when it comes to genetic therapies, contacting me if you need help with navigating trial participation, and just utilizing all the resources that you have. But again, patience is really going to be the biggest thing.
Our next question is, are there any known side effects of the genetic therapies?
Yeah, so I'll start. I highlighted a few of these from the trials that we've seen. So they can include fevers, headache, nausea, exacerbation. The 4D trial did report one patient with severe lung inflammation. So we know that these are things that appear in the trial. We don't know what the side effects would be from an approved therapy that you take all the time. In the case of mRNA therapy, gene therapy would be a single dose, but those are what's been reported so far.
Next question is, will you please discuss Prime Medicine and what their research scope is and what is different from other genetic research in CF?
Yeah, I can answer that. So Prime Medicine is a gene editing company, and we recently released a new investment in their technology, which utilizes a newer type of gene editing called prime editing, therefore the name Prime Medicine. One thing that's particularly exciting about prime editing compared to traditional CRISPR, and I'm getting a little into the weeds, traditional CRISPR creates what we call a double strand break, or a cut through both strands of DNA. And we've learned in recent years that this can actually have a lot of side effects inside cells and cause what we call genomic instability, or other cuts and breaks not at the target location. prime editing does not make a double-stranded break. And so it's thought that it could be a safer gene editing approach. There are no prime editing therapies out there yet. So this is a discovery stage program that will look to edit some modulator ineligible mutations. And we are super excited to see where it goes.
Awesome. That's so fascinating. It has been reported that Vertex may be seeking a priority review voucher for early approval for the, I believe, new Vanzo triple combo. Can a PRV help advance approval for these genetic therapies?
I don't know the answer to that question.
I do not either.
Super fascinating. Yeah, it's so new, I feel like. Oh, this is another fascinating question. Are these therapies effective for symptomatic holders of a single gene?
So from a biological perspective, they could be, but these clinical trials will enroll people with an official diagnosis of CF, and the approval will be for those with a diagnosis of CF. So right now, there's not really a way to explore that other than like physician-initiated research. So without the CF diagnosis, drugs for CF wouldn't be able to be trialed or tested.
Hey, I think we are probably about ready to wrap up and have our final question, but I think it is a really great one. From a scientific perspective, how hopeful are you feeling? Excuse me, toward finding a cure in your lifetime?
I don't know if that's, or I can start. Katie, maybe you have an answer too. I'm excited. I think cure means something different to everyone. And I kind of, in my talk, mentioned when I think about it from a scientific perspective, that's correcting the CFTR gene in all of the affected cells and organs. I think that's going to be a real challenge. I would definitely hope to see it in my lifetime. I do think we will see a genetic therapy for the lungs in the next, I hate to keep saying 10 to 12 years, but it'll still be time. But yeah, I'm really hopeful. The science is moving fast and we're learning more every day. So I have a lot of hope that these will come to fruition.
Yeah, I'll reiterate that. There is a lot of hope. Being able to see Trikafta and modulator therapy go from early phase trials since I started CF research to FDA approval, these trials, the science is moving fast. And so I'm very hopeful that it will happen at some point in time.
Yeah. And if I can add one more thing, it's just that we're not stopping. So just because there's mRNA trials in the clinic doesn't mean we're not looking for new RNA technologies or different delivery vehicles from AAV and LNP. So we continue to invest in those early stage technologies because we want to guarantee that we find what is right for CF.
Perfect. Well, thank you both so much. I know I definitely appreciate all the info that you've shared this evening. And thank you everyone so much for coming. I will just close here to remind everyone that the recording will be posted on YouTube within the next few days, and I believe it will be shared via email. And we also do have an upcoming opportunity to support the members of the CF Foundation. Let me just get my info here once again. I'm so sorry, everybody. But it's essentially going to be hosted by the CF Foundation or like a group of people with CF. I'm so sorry, I'm completely messing this up. But essentially it's going to be a discussion about people who are either not eligible for modulators, or maybe like myself, you are eligible but not currently taking a modulator, maybe due to side effects or whatever have you. And just kind of having a small group discussion about that. And I believe more info is going to be added in the chat there and in the follow-up email for this presentation once we're done here, but I will wrap it up since it is just a minute over our time here. So thank you both again. We definitely appreciate you. And thank you everybody